Hi, my name is Peter Andras. I am a professor at Edinburgh Napier University, and this is my lecture series on evolution of complex systems. This is lecture 4, Cells and the Genome, and this is part of lecture 4. So, in this lecture, we will look at the genome, seeing it as the information subsystem of the cell, discuss about the genome as a system, how the identity of the cell is defined, and how identity violations and adaptations in cell happen, how the cell responds to these, and how we interpret the complexity of the cell and of the genome. So, as you may remember, in the previous part of the lecture, we looked at the cells, and we said that one way to discuss the cell is that you look at the components, the structure of the cell, and describe what you find in the cell. The alternative way to look at the cell is that you see it as a system of protein interactions, where the proteins and other molecules interact and deliver through these interactions the full range of functionality of the cell, where these molecular interactions are organized in space and time such that these functions are delivered. And we said that if we look in this way at the cell, we can see the cell as a system of molecular interactions, and in particular as a system of protein interactions. Now, at the end of the previous part, we noted that the mRNAs can be seen as memories of protein interactions in the way that they represent the memory of interaction between one amino acid and the string of amino acids. And we also said that the DNA gene segments, so the parts of the DNA that encode the genes, can be seen as memories of RNAs and of RNA interactions, in the sense that the transcribed premature RNA includes many short regulatory sequences which get spliced out from the premature RNA and interact with the rest of the RNA to produce the mature mRNA in a context-dependent manner, where the context is set by other RNAs present in the nucleus. Just to note that all these interactions which involve RNAs and DNAs are typically supported by proteins. So, for example, the transcription of the RNA from the DNA is supported by the RNA polymerase and the helicase, which opens up the DNA double helix. The spliceosome proteins are the ones which are involved in the splicing of the RNA. It is noted that there are some self-catalyzing RNAs which can splice out themselves from the premature RNA, but in general, the involvement of proteins and the molecular interactions with proteins are important. Now, what we say here is that we have a lot of interactions between the DNA segments, RNAs, and of course proteins involved, which regulate which parts of the DNA get transcribed, which are the active parts of the DNA. Now, just to note here, there are also a lot of other proteins involved here which trigger the activation of some parts of the DNA, transcription factors. And there are other interactions between various parts of DNA as well. So all these together can be seen as the molecular interactions that constitute the information subsystem of the cell. This is because, as we said, the DNA segments and RNAs represent, at least some of them represent memories of interactions within the cell. So they are information representing molecules, and processing these constitutes the information subsystem, as we discussed in general in the context of this systems theory. So Overall, the genome can be seen as, of course, a description of all proteins produced, but also a description of all the regulatory interactions that are needed to produce the right RNAs at the right time, which turn into the right kind of proteins through the translation process within the cell. So in this way, we see the genome as information subsystem of the cell, also the subsystem that defines the identity of the cell. So what we provide here is a redefinition of how the genome defines identity of the cell in the context of this systems theory interpretation of cells. Now, if you look at particular cell types, for example, prokaryotes, like bacteria or archaea, what you see is that they don't have a nucleus. Their DNA is organized in kind of circular structures, possibly quite long and no longer circularly organized. Now, what we see here is that although there is some level of compactification of the DNA, there is a coiling of the DNA, and histones are around which package it to some extent. This separation from the rest of the cell is much less present in prokaryotes than in cell-cis nuclei. Also, what we notice is that in the prokaryotes, a large part of the genome is actually a coding genome, quite often over 75% of the genome. And the part of the regulatory component of the genome is relatively small. So there are many interactions between the DNA and proteins within the cell in general. So the information subsystem is somewhat less separated off from the cell. Now, on the other side, if you look at eukaryotes, so these are the cells with a proper nucleus, what we see is that the genome is compacted in the nucleus. There is a physical separator, the nuclear membrane, which separates the nucleus, the inside of the nucleus from the cell cytoplasm. And clearly here we have many more interactions between the RNAs, DNAs, and the supporting proteins, which is enclosed, much more separated than the case of prokaryotes. And also we have a much larger part of regulatory content in the DNA. The regulatory content can be over 95% of the DNA. So let's look at the genome. As you may know, you have 4 nuclear bases: adenine, cytosine, guanine, and thymine, ACGT, which compose it. These are attached to phosphorylated sugar molecules, and those form a backbone to which these various bases are attached. Only these 4 bases are used. And in the RNA, you have a single backbone and a single series of bases. There, the thymine is replaced by the uracil. And in the DNA, you have a pair of string of nuclear bases. The pairs are organized in matching pairs. The A matches with T, the C matches with the G. And there are hydrogen bonds between them to keep the structure more stable. The structure is organized in a helical way, and then it is called up further more in many ways to make it much very compact structure. So a very long sequence can fit into a very small place within the nucleus of the cell. Now, the DNA itself contains coding segments. These are traditionally called genes. And non-coding regulatory segments. Historically, there was the assumption that there is also junk in the DNA that is not used. However, more recently, it seems more likely that actually all parts of the DNA are meaningful and useful. Although some of it we just don't know what they do, but it seems that they all contribute either to the coding of proteins or regulation of how the coding is translated into or transcribed into RNAs and then translated into proteins, or contributes to the maintenance of the structure, stability of the DNA, or supports the replication of the DNA. So, as the cell works, different proteins are needed for the normal functionality of the cell. So how this happens is that different parts of the genome are activated at different times, and these are given as regulatory networks of genes. So some gene gets switched on that triggers the switching on of some other genes and so on through the interaction with the regulatory components of the DNA. Typically, there is a transcription factor protein which comes from the cytoplasm, triggers the switching of some part of the DNA, and then a sequence of further switches follow on according to the regulatory network to which this part of the DNA belongs. So this means up and down regulation in a dynamic way of the amount of proteins produced within a cell, and this change in the proteins that are produced within the cell supports the changing behavior and the changing needs of the cell. So what we can see here is that we have sequences, or in more general sense, patterns of interactions of molecules involving genes and regulatory segments in the DNA, and also RNAs and proteins that are required. And these patterns follow each other. So we mentioned already gene regulatory networks. Another important and relevant element is the topologically associating domains. So these are parts of the DNA that are organized in space such that they are nearby to each other. They form kind of phase-separated liquid bubble in the within the nucleus, where these parts get co-regulated and effectively support the gene regulatory networks and the joint regulation of the transcription of a number of genes, and also of regulatory content. So what we see is that these patterns of interactions follow each other, and we can see these patterns of molecular interactions as the communications within the genome. Now clearly, one set of molecular interactions involving the DNA depends on other molecular interactions involving the DNA. So in this sense, so for example, when the transcription factor initiates the activation of a part of the DNA, for example, by activating the helicase, which opens up the DNA segment for transcription. Now, this is how the patterns of molecular interactions reference other patterns of molecular interactions within the DNA. So to have a certain pattern of molecular interaction, you need to have a set of other molecular interactions before. So in this way, we can define the referencing of earlier communications within the genome by future communications within the genome. So what we see is that there are many interactions within the genome involving the DNA, various RNAs, and some proteins. And there are relatively few interactions outside of the genome or outside of the nucleus in the case of eukaryotic cells. So these interactions outside which are relevant are, for example, the molecular interactions that produce transcription factors, or the molecular interactions that happen with the mRNAs which are released from the genome, from the nucleus. So these are relatively few interactions compared to the number of interactions that happen inside of the nucleus, or in the case of prokaryotes, with the DNA distributed across the cell. So what this shows is that there is a clear system boundary in terms of the frequency of molecular interactions. This density boundary is much more clear and much more steep in the case of eukaryotes, but there is a boundary in the case of prokaryotes as well. So now that we define the genome as a molecular interaction system, let's have a look at what are the actions and perceptions of this system. So on the actions, we need to look at molecular interactions which impact the environment of the genome. And this environment, the immediate environment, is the cell itself. So these impacts are really by delivering RNA molecules into the cytoplasm. These are mRNAs, tRNAs, RNAs, and a few other kinds of RNA molecules, which are active outside of the nucleus. And ultimately, these produce or support the production of the proteins within the cell. Now, the production of these molecules is of course referenced inside of the genome by further molecular interactions within the genome. But they deliver impacts on the environment of the genome within the cell. Now, looking at the perception side, so these are perceptions about the environment of the genome. Again, the environment in this case is the cell itself, the cytoplasm of the cell. And these perceptions are changes in the molecular interactions within the genome relative to expectations of these molecular interactions. So, for example, when transcription factor molecule, a protein, interacts with the genome, that changes how the genome components get activated. So there is a difference between what was expected based on prior molecular interactions, and this represents a perception of the genome about its environment, which is a cell itself. Of course, this leads to new generation of new genome communications, which of course turn into the production of some RNA encoding, protein encoding RNAs, and also may turn into the production of some other protein encoding RNAs. So you can see the genome overall as a system where the molecular interactions involving parts of the DNA and RNAs constitute the genome as the information system of the cell, and this defines the identity of the cell in the sense that it defines what is acceptable and non-acceptable communication within the cell. So the genome the interactions, molecular interactions involving the genome can be fitting or not fitting the identity of the cell, and this is decided through many interactions of these molecules, and it also leads to the activation of the correct mRNAs, tRNAs, and so on, which produce the right proteins within the cell. But we'll note in a moment meaningful and meaningless interactions are. But just note here is that, for example, if you have some virus which gets incorporated into DNA, that might trigger inconsistent communications within DNA relative to the identity of the cell, or transposons or retrotransposons may trigger such inconsistent interactions. These are parts of the DNA that can jump from one place to another or can copy themselves from one place into another place within the DNA. Overall, as a system, the genome's aim is its own maintenance and expansion. The maintenance involves, again, lots of protein interactions with the genome, which aim to fix any problem that emerges in the genome. Now, these problems could be, for example, during the transcriptions, sometimes the DNA may get broken. Sometimes some of the letters may get lost in that process, so these need to be fixed. There are also other possibilities, for example, irradiation by high energy rays, like gamma rays, for example, X-rays, may cause changes in the genome, which again needs to be fixed. So all this is done by a number of protein interactions which help the genome to fix itself. On the expansion side, of course, the genome aims to expand. So if we look at the genome expansion, essentially what we find is the replication of the genome, which induces cell division and an expansion of the cell itself. Now, the replication involves the full replication of the genome. Again, it may involve some errors in the replication, which again need fixing. It leads to the expansion of the cell. However, there are cases of cells where there are cells with multiple nuclei. So that raises the question of how does it fit with all this discussion of having a genome defining the identity of the cell, but having multiple nuclei in a cell might define multiple identities, although these might be very close since they are copies of each other, at least potentially. Now, examples of these are, for example, muscle cells, or liver cells, or cells of some fungi. And there are other examples as well. So what we see in this case is that from the perspective of the systems theory, what we see is that the different nuclei are active in different parts of these cells. And effectively, they deliver the identity system service to a part of the cell which is localized around them. Although there are no clear structural dividers between these parts of the cell, what we see is that in some cases there is some level of structural divisions, for example, in muscle cells, or in the fungus cells, or for example in the embryo of flies. In the early stages, the zygote is not dividing into separate cells, but creates multiple nuclei, which are located in different parts of this large cell, which is subdivided by some membrane structures, but these are not the effective cell membranes. So overall what we can see is that in multinuclear cells, there is some level of separation between different parts of the cells, although that separation does not constitute the cell membrane. So different parts have relatively different identities, although again, quite often the expectation is that these different identities are very closely aligned, so the differences are relatively minimal. So, what we discussed so far shows that the genome is seen as a system of molecular interaction communications, where the language units are these regulatory interactions of the genome segments, which lead to the activation of some of the segments that ultimately transcribe into mRNAs. The grammar is seen as the conditional rules which tell the probabilities of what may follow previous molecular interactions. And we showed that the referencing rules are the ones which tell you what previous molecular interactions in the genome are required for a certain molecular interaction to take place. And the continuation rules tell you the other side, given a set of molecular interactions that take place, what are the possible continuations, what other molecular interactions may take place, and what are the probabilities of these possible continuations. So all these together constitute the language, the system language of the genome. Now, to define what is meaningful and what is meaningless in the context of these communications, we follow the discussion that we introduced in the general systems theoretical lecture. So the meaningful molecular interactions are the ones which have continuations and which fit the identity, fit the rules of the communications within the genome. The meaningless communications are the ones which do not fit these rules. So, for example, molecular interactions involving a copy of a viral DNA or some mutated DNA, which lead to the production of proteins which are not fitting the cell, possibly interfere with cellular processes, and potentially can damage the cell and kill the cell ultimately. Now, we noted the viruses already a couple of times. So, if you look at the viruses, what you see is that this is either pure DNA or RNA in a lipopeptide envelope, and that possibly is covered by further a lipid membrane, and it may have a number of sugar molecules attached to it. Now, these viruses attach to cells, their DNA or RNA gets released into the cell, and there it interacts with molecules there. Sometimes it interacts with only RNA molecules, sometimes it gets incorporated, enters the nucleus and gets incorporated into the DNA, for example, the herpes virus. And there are many other cases where viruses work in hard to understand ways. For example, in plants, there are viruses that are passed from one cell to another only through cell division, so the virus never exits the cell. So there are a range of these viruses. And the question is, what do they do, or how could we interpret them in the context of the systems theoretical analysis of cells? And what we see here is that they can be seen as cell memories, which are passed on from one cell to another and interact with the cell memories that are in the cell. Now, we know more about the cases when these interactions are damaging, so for example, illness-causing viruses. But clearly there are many other viruses which do not cause illnesses. As I mentioned, in the plants, there are some viruses which are passed on from cell to cell during cell division, and potentially may have some function in these cells. There are at least a few cases where there is some indication that having viruses adds to the functionality of the cells. For example, in some algae, some of their viruses bring in encoding of molecules which are not encoded by the cell and helps the functionality of the cells. However, overall, it is quite unclear what would be the purpose of these viruses. We can interpret them in the context of the systems here as memories of molecular interactions which interact with the memories within the cell, and potentially these interactions might be useful and might be also harming to the cell. Clearly, in the case of viruses that cause illness, the interactions are quite harmful and can often lead to the death of the cells which get infected by the viruses. And correspondingly, cells have antiviral defenses which are aimed to stop this negative impact of the viruses on these cells. Now, faults, we said that these are communications which do not fit the language. So, for example, when a cell, a bacterium, is put in an antibiotic environment, those antibiotic molecules interfere with the protein interactions of the bacterium, causing wrong interactions, and in that way damage the bacterium. Or the interactions with the viral genome may be very damaging, causing the wrong kind of molecular interactions with the cell. The errors are the situations where communications are following the rules, but there is no continuation. The continuations are expected, but they do not happen. So, for example, if a cell, let's say a bacterium, is put in an environment where some nutrient is lacking, the cell is producing the right kind of molecular interactions aimed to pick up the nutrients, but the nutrients are not there. So the expected continuation molecular interactions do not happen. And finally, the failure is the case when there are many error situations and many faulty communications within the cell, and that lead to the shrinking of the cell and potentially to the death of the cell. So, for example, antibiotic exposure in the bacterium or radioactive damage to the genome, which causes the production of wrong proteins in the cell, may lead to failures and to death of the cell. Now, in response to the identity violations that the cell is experiencing, there are adaptations that the cell is producing. So the cell perceives the environment through these identity violations and aims to eliminate these identity violations by changing the protein interactions that are present in the cell. So, for example, if the nutrient environment of a bacterium is changing, the bacterium may trigger the production of a different set of proteins which adapt to the new environment and pick up other kinds of nutrients that are present in the environment or facilitate the pickup of these nutrients. Or, for example, if you look at a neuron, when the neuron receives a certain pattern of spike input, that also triggers the activation of proteins and signaling pathways, which may trigger the activation of some transcription factors in the nucleus, triggering the production of the appropriate channel and receptor molecules so that the neuron can respond better to these inputs. So, for example, if you look at the dynamics of dendritic spines, where the synaptic interactions happen with other neurons, these spines can grow or shrink depending on the input that they receive. So In general, the adaptation happens following the perception and then generation of molecular interactions that lead the action of the cell onto the environment, such that the, the proteins that are produced in the cell adapt to the perceived changes and aim to improve the fitness of the cell within the changed environment. Now, as I already mentioned, the changes in the cell may involve changes in the activity of the genome, and these are the adaptations at the genome level. Some of the changes of the cell are passed on through the use of transcription factors, and these trigger the activation of different parts of the genome, producing different kinds of RNAs, and then ultimately producing different kinds of proteins within the cell. There can be also other identity violations within the genome where wrong molecular interactions happens, possibly because of incorporation of viral DNA, possibly because of reverse transcriptases, possibly of other reasons which change the DNA, and again these trigger molecular interactions that aim to correct these. So for example, if there are changes in the letters that do not fit within the DNA, there are repair mechanisms involving a number of proteins which fix these changes and correct the errors in the DNA. Now, just note that there is also some evidence of RNA-mediated corrective adaptation in the genome. There was, for example, an experiment on the Arabidopsis plant where they showed that the genome of the plant cells was changed after some generations. They could return to the normal production of the normal parts of the plant. And the assumption was that this was mediated by RNAs, and using the reverse transcriptases, which translate back RNAs into DNA. Other evidence is not fully explicit. This was assumed and inferred from the data. So this is a possibility of fixing changes that are wrong changes in the DNA. Another adaptation aspect is the emergence of antibiotic resistance in bacteria. So this happens through mutation and selection, and also recombination with other bacteria. So in this case, the presence of antibiotics in the environment triggers adaptations in the DNA of the bacteria, which lead to the production of molecules which confer antibiotic resistance, for example, by breaking down the antibiotic molecules or shuffling them out very quickly from the cell. Again, in this case, the identity revision, the change of DNA happens by mutation and selection, and possibly by picking up elements of the DNA from other cells. Finally, let's look at the complexity of the cells. If you look at prokaryotes like bacteria or archaea, these are simpler and produce lower amount of biomass. On the other side, eukaryotes are much more complex. They have much wider range of behaviors, and they can adapt to a wide range of environment. They are also much more compactly organized, much more densely organized, and then they produce a higher amount of biomass. So what we see is that the cells with nucleus are much more complex, much larger, where the cells without nucleus are simpler compared to other cells. The same way, if you look at the genome, the amount of regulatory content determines the complexity of the genome. The ones which have relatively less, small amount of regulatory content, they have less adaptivity built into them, while the ones which have a large volume of regulatory content, these cells can adapt to a wide variety of environments, and in a sense they have better actions and perceptions reflecting better their environment. So, to summarize, we looked at this part of the first lecture at the information subsystem. We said that this really involves the DNA and RNA molecular interactions, also involving a number of related proteins. And we said here the communications are these interactions involving the DNA and possible RNAs. The language itself is made of referencing and continuation rules, where referencing rules say which molecular interactions can need to happen before certain molecular interactions becomes possible, while the continuation rules indicate what molecular interactions may happen after a certain set of molecular interactions happened in the past. Just to note that in principle this language could be known. In practice it is not, because we just don't have enough experimental data to determine this kind of language. So we define the genome as a system of molecular interactions involving DNA and RNA, and we said that this system is the information subsystem and also the identity defining subsystem of the cell. Then we discussed about actions and perceptions of the genome system. And they said that actions are the production of the mRNA molecules and plus the other RNA molecules which are released in the cytoplasm of the cell, and their impact what kind of proteins are produced within the cell. While the perceptions are, for example, the activation of parts of the genome by transcription factors, or activation of other molecular interactions which interfere with the genome following the perception or following the changes from the expected interactions. So, for example, these lead to the fixing of the DNA following damage suffered by the DNA. Then we discussed about meaningful and meaningless communications, and we said that meaningful are the ones which lead to further continuations that fit the rules, and the meaningless are the ones which do not do this. For example, they occurs because some part of DNA is damaged by radiation, or because the incorporation of viral DNA or viral RNA present in the cell. And finally, we discussed about complexity, both of the genome and the cell, and we said that the cells which have nucleus are generally more complex than the cells which don't, and cells which have a genome which contains more regulatory content are generally more complex than the ones who have less regulatory content. Finally, let's have a look at a number of questions and answers. First question is it true that the genome is the information subsystem of the cell? And the answer to this is yes. This is what we said, that the genome includes all these molecular interactions involving the DNA and the RNAs, and this manages the information about the cell, which is contained in the RNA and DNA molecules, which represent the memories of the cell. Second question, is it true that the generation of an mRNA is a result of referencing to other genome communications? And what we said is that indeed this is the case, because the generation of an mRNA is the result of many molecular interactions involving RNAs and proteins, which follow a number of molecular interactions that involve the DNA and RNAs and proteins. So in this sense, the production of an mRNA is a result of the referencing of other genome communications. Third question, Is it true that mRNAs represent the perceptions of the genome system? And the answer to this is no. As we discussed, the mRNAs are rather the actions of the genome systems. These are impacts of the genome system on its environment, which is the cell itself, the cytoplasm of the cell, while the perception are about changes within the genome communications that are induced by the environment. So the mRNAs are not the perception, but rather actions of the genome system. Finally, is it true that the language of the genome system is defined by grammatical rules representing conditional probabilities of genome communications? And the answer to this is yes, indeed, that's what we said, that in the context of this systems theory interpretation of cells, this is the language of the genome system. This is how it is defined by these grammatical rules. However, what we also said is that although in principle we can say this, in practice we do not know these grammatical rules because we just don't have enough experimental data and the sufficient kind of measurements that would allow us to decipher which are these grammatical rules. Thank you.